In the second part of the blood chapter presentations, we're going to look at the components of blood, which is to say the major components that make up the tissue of blood, the cellular and the extracellular components. And we're also going to consider the clinical test that separates those components out as a measure of how much of the blood has oxygen carrying capacity. When we consider what makes up blood as a tissue, there's two major components that we're going to look at. And then I want to look at a few specific molecules within one of those, and then consider how we separate those components out in a clinical test. This table is going to show us the different components that make up blood and provide some figures representing how much of the blood is made up of each thing. There's a couple of things I want to mention about this table to begin with. First off, I'm never entirely happy with the numbers that textbooks provide for this. This textbook is a little bit better in most regards in that sense, but for this one and with any look at blood, I'm not terribly crazy about the seeming specificity of the numbers. For some of these things, they should be rounded off a little bit more. Instead of saying the plasma is made up of 92% water, it really should be rounded off to a more general number, like 90%, for example. I'll talk a little bit about that in a second. So the first component to talk about is the plasma. That's the extracellular component of blood. And blood is a connective tissue. What really defines connective tissues is what makes up the non-cellular or extracellular space. And for blood, that's plasma. Now, I mentioned the specificity of numbers to some degree. Now, I like in this book that they give ranges more often than averages. But, like I was saying a second ago, to call the plasma 92% water is actually, I think, a little bit too precise of a number. It really should be 90%. Now, in science and mathematics, there's a concept of significant figures. So when we look at 92, that's saying that the value there is somewhere between 91.5 and 92.5. But if we say 90, then that means that the value is somewhere between 85 and 95. Now, it wouldn't really range quite that much, but it's more appropriate, I think, to say 90% and understand that there's a bit more looseness in the numbers. So I'll usually think of water as being 90% of what makes up plasma. Now, plasma is also usually around 55%. It does vary quite a bit from person to person, but if we think about plasma being 55% of the whole blood and water being about 90% of that, then that means that blood is about 50% water. Now, the other things that we see in the plasma are plasma proteins, which are very important, and then some regulatory proteins like hormones and those sorts of things, and then other things, small molecules that are dissolved in the water. The numbers given there for what part of the blood they make up is fine. I'd adjust them a little bit just to reflect the 90% figure for water, but that's fine. The plasma proteins are albumin, the globulins, and fibrinogen. Now, those aren't all of the proteins in the plasma. Those are just the three major ones. Other proteins are found, the regulatory proteins, for instance, at the bottom. But these three make up the most of that. Now, albumin's almost 2/3 of the protein in the blood. Its major reason for being there really is to take up space, to make blood thicker than water. It also plays a part in helping lipid molecules be transported, like steroid hormones or cholesterol or the thyroid hormones, like we talked about in the endocrine system recently. globulins are a few different types of proteins. They make up almost a third of the proteins in the plasma. And there are various types, but collectively they are involved in, again, taking up space in the blood, making it thicker. Then also there are immunoglobulins, which are also known as gamma globulins, and those are more commonly known as antibodies. Fibrinogen is the major fibrous protein in the blood that's related to blood clotting, and we'll talk about clotting separately in another section of the presentation. Now, a little bit of a word about some of the other solutes. They're small molecules, nutrients, gases, and wastes, as the table here says. And if we think about gases, usually we think oxygen and carbon dioxide are the major ones. But also nitrogen is in our blood, and that's because there's so much nitrogen in the air we breathe it gets into our blood, but it's a molecule that we can't break down, so it's inert as far as our body's concerned. We breathe it in, we breathe it out. It doesn't do anything other than get in there. It's best known for being the cause of getting the bends if somebody surfaces during scuba diving too quickly. And the concentration of nitrogen in the air that is being breathed through the scuba tank and the air outside aren't equilibrated a little bit. That's going to cause big problems for the gases that are in the blood. This is an artist rendition of the albumin molecule. Now, the artist in question here is really a pretty incredible guy. I've met him personally. He's a biochemist that works out in La Jolla, California, which must just be a cool place to live. But he works in looking at the structure of proteins for his research, but then also he interprets the representation of protein structure in art like this, which is also educational in purpose. He works primarily in watercolors, and so he makes use of color to look at molecules like this. Now, this is albumin, and you can see from its overall structure that it is fairly globular, meaning that it doesn't have a whole lot of real shape to it. It's just, taking up space. He's chosen to represent the structure of this instead of with the classical colors for chemistry models, to instead make the protein portion, the carbon, oxygen, hydrogen, nitrogen atoms that make up the amino acid chain, sort of fade into the black background. There's a little bit of variation in color to illustrate that there's different atoms making up the amino acids in this protein. But what really jumps out in the picture are the little red dots, and those represent oxygen atoms on probably fatty acids. It could be some other lipid, but the point is that lipids fit into little pockets in the albumin molecule. So while it is part of making the blood more viscous, it also has just slots for lipids to slip into and move through the blood that way. This is another molecule that the same artist represented. And by the way, underneath the picture of albumin and underneath this picture of an antibody, there's a link that goes to the protein database website. And he maintains a very important part of that, which is called the Molecule of the Month. It's a great site for educational purposes to highlight the structure of various molecules, mostly proteins, although there's DNA and some other things listed there. But he does a great job of doing that. So this is his representation of an immunoglobulin, or an antibody. Now, an antibody is made up of 4 separate protein or polypeptide chains. 2 of them, which are represented in the reddish and orange colors in this picture, are called the heavy chains, which are the longer ones. And then the 2 that are represented in yellow here are called the light chains, or the shorter ones. It sort of takes on a Y shape, and often you'll see schematics of antibodies as a Y, with the arms of the Y being the light chain and part of the heavy chain linked together on either side. And then the two heavy chains where they come together are make up the tail region. Now, I don't know that anybody necessarily refers to those structures with those terms, but that's the way that I usually describe them. What's really important is where it thins out between the two is a hinge region that gives the molecule some ability to flex at that point. This is a figure from the Encyclopedia Britannica website showing a little more detail schematically of how antibodies work. So in the top right corner there's a graphic representation, schematic representation of the antibody. In light blue are the heavy chains, and in pink are the light chains. Now where they come together, the pink and the blue side by side, are the two arms of the Y-shaped structure. And you'll see some notches cut out of that. That represents what's called the antigen binding site, or variable region of the antibody. And that's how it interacts with things that can get into our body. And then there's more of a spring-looking representation of that hinge region that I mentioned a second ago. But it does allow for the two arms of the Y to move around a little bit. So they have an antigen here, just some spherical body with some things sticking off of it, which might be a virus particle or something to that effect. And we see first an antibody binding to two spots on that, and those spots that it's binding to are called antigenic determinants. They're protein structures on the surface of an antigen that an antibody physically binds to, like puzzle pieces coming together. And you can see the shape of the antigenic determinant in this cartoon complements the shape in the antigen binding site region at the end of the Ys. And then we see one antibody linking two separate antigens together, binding to an antigenic determinant on either one. And then one of those antigens is bound to another antibody, which binds to another antigen, linking all of those together, making them into a bigger mass of things that can then be cleared away by white blood cells and hopefully not be virulent, which will help protect us from whatever disease these might suggest. So the other portion of the blood is called the formed elements, which is kind of a fancy way of saying the cells that are made specifically for the blood. Now, there are a couple little problems with the table that I want to point out here. First off, the erythrocytes, or red blood cells, are in a row that's unfortunately at the bottom of the plasma part, not the top of the formed elements. So the line separating formed elements and plasma in this table is just in the wrong place. Second, I've tried to cover up where they duplicated some information. Under where it says leukocytes less than 1%, it actually says platelets less than 1%, and then it says platelets less than 1% in the bottom row also. So just an odd little duplication there. Now, erythrocytes, or red blood cells, make up 99% and then some of the cells in the blood. It's largely what's there, and they're responsible for transporting oxygen primarily to all the cells, as well as clearing carbon dioxide away. Leukocytes are white blood cells. There are a couple of different types, granular and agranular, and there's subtypes within those. They're mostly responsible for immune function. Some of them nonspecific, some of them specific, but they're immune cells for the most part. And then there are platelets. Now, I might have put in that blank spot there next to where it says platelets less than 1%, maybe the Greek word thrombocyte, which is another word for a platelet. Now, platelets are really cell fragments of a larger cell called a megakaryocyte. Now, that doesn't mean that it's not part of the cellular component of blood. It entirely is. just understand that they are really little things, and it's just the megakaryocytes broken up, and a little bit of each platelet has some of that original cell in it, and their job is for forming blood clots. Now, this little table of information about the numbers of cells, I added in here, and I just think it's a good way to consider what these cells are about. Now, the top numbers are the numbers of cells that would be found within 1 microliter of blood. Now, the program I'm using to write this in doesn't allow me to put in Greek characters, so I'm using the lowercase u instead of the Greek letter mu to represent microliter. So within that volume of blood, which is very, very small, a drop of blood would be 50 microliters on average. So it's a very, very small amount of blood. There would be 5 million red blood cells on average. And then there'd be about 250,000 platelets. And at the upper end of a healthy person, 10,000 white blood cells. Now, I'm using those numbers because they're nice and round and they fit into the ratios I'm about to share with you, but really they represent a range somewhere. Red blood cells are usually between 4 and 6 million. Platelets, I think, are 150,000 to 400,000, and white blood cells in normal situations should be between 5 and 10,000. Lower than 5 is immunocompromised. Higher than 10,000 is fighting off infection. Now, just to illustrate a little bit of the relationship between these types of cells, for every red blood cell, for every platelet, there's going to be 20 red blood cells. And for every white blood cell, there's going to be 500 red blood cells. And those ratios come from the numbers that I'm giving you at the top, just because they fit together so nicely. Now, I want to discuss a test that would be used in a clinical setting called the Hematocrit. Now, what happens here is a sample of blood is taken. In these pictures, this represents a 10 milliliter vacutainer of blood, for instance, your standard blood draw kind of container. And the blood is spun around in a centrifuge to separate out the different components. Some will go to the bottom of the tube and some will stay on the top of the tube. The effect that causes this is very similar to why oil and vinegar in Italian dressing separate out. Okay. Now, in doing this, you can compare the percentage volume of just the red blood cells part with the total volume, and that'll give you the hematocrit number. Now, the numbers at the bottom there represent normal ranges for men and women, or women and men. The lower range, 37 to 47, is for women, and then the higher range, 42 to 52, is for men. The other two tubes that we see here represent a couple of other situations. Anemia is really a lack of oxygen carrying capacity, and so if you have too few red blood cells, you're going to have anemia. Now, it's possible if the percentage is just at the low end of the normal or just below the low end of the normal, the oxygen carrying capacity can be offset. If there's too few red blood cells, each red blood cell could have more hemoglobin in it and therefore be able to carry more. The last tube represents polycythemia, which is literally too many blood cells. And in this case, the plasma is low and the blood cells is high. This is often the result of something that stimulates overpopulation or an overgrowth of red blood cells. But it just as easily could be something that you'd see in dehydration, too little water in the blood. In this situation, the blood's going to be very thick. All these red blood cells are going to gum up the blood and make it harder for it to move through circulation. Consider this question when you feel that you've reached an answer hit the next button to go into the next slide, which will reveal the correct answer. Now, the hematocrit is a measure of what percentage of total blood is the red blood cells. And that means if it's 45, the plasma would be 55% of the blood. It'd just be the opposite of the hematocrit number. So if somebody's drinking during the day, they're going to be adding water volume to their body and to their plasma, which means that the plasma volume will go up relative to the total blood volume. And that means that the hematocrit number will decrease. And it's not unusual to see this. Somebody might drink over the day, and it can actually change the hematocrit number by a few points if enough water is ingested over the day.